Sam is a research landscape ecologist at the U.S. Forest Service Rocky Mountain Research Station in Flagstaff. He's worked as a Forest Service scientist since 2003 and has been here in Flagstaff since 2009, where he is an affiliate faculty in the School of Forestry. Sam's research focuses on a range of topics in landscape ecology, including species distribution modeling, landscape genetics, climate change, fire ecology, connectivity modeling, and applications to guide management and biodiversity conservation. He earned his BS at the Evergreen State College, MS at Western Washington University, and his PhD at the University of Massachusetts. Let's give a warm welcome to Dr. Sam Cushman.
It's good to be back in person after all this remote Zooming. My first live lecture. So that's good. The talk today is going to be about that maybe. Integrating movement data, genetic analysis, to understand connectivity of animal populations in the context of conservation. And just as a beginning, some big principles that put it into context, right? We've probably all heard of the theory of island biogeography, species area relationships, extinction thresholds as a function of habitat area. One of the main principles of spatial ecology is habitat extent is important. Population size, population viability are higher when there's more habitat. And there's non-linear thresholds where extinction risk increases greatly as habitat extent shrinks, related to dispersal ability, demographic potential, habitat suitability. So that's a classic foundation. And then we started to talk about fragmentation. That came after the island biogeography stuff. Fragmentation started up in the 90s, really. People started to care about fragmentation. And they concluded that in many cases, breaking habitat up into different blocks was a bad thing. And having the same amount of habitat in one big connected patch was better than breaking it up into little patches. So generally, habitat fragmentation can increase extinction risk more than you would expect just based on habitat loss. And fragmentation effects are usually bigger for less mobile species, except when fragmentation influences mortality risk, in which case the fragmentation can increase mortality more for the more mobile species. And then it's not really a connectivity problem, it's a mortality risk problem. But fragmentation, generally a bad thing in most cases. And then in the 2000s, we started to talk about corridor connectivity, which now is even now at the peak of its popularity as a conservation subject. The idea is fragmentation is bad, but we can mitigate it, right? We can put corridors between these fragments and that will make them connected so that they will no longer break this population up. And it may improve connectivity in some cases, depending on if those corridors are well placed, and well proposed, so that they're functional in promoting movement or reducing mortality risk. But we've also learned since then, in a lot of recent stuff, coupling simulation and empirical data, that improving connectivity to things like corridors generally does not mitigate habitat loss, right? So if you take half the habitat away and then put bridges between the remaining patches, it's still not as good because those bridges don't replace the lost habitat. They might increase movement, reducing extinction risk, maybe promoting genetic exchange, but generally linkages and connectivity does not replace the effects or mitigate the effects of habitat loss. All right, so connectivity, key to viability. So here's a picture of three things that really guide the drive for connectivity. First, in the background, you see a map, right? That map has dark blue areas, it has light blue and gray areas. We call that a resistance surface, right? That's a map that shows the cost of movement to every pixel in the landscape. I'm going to get into how this is made to fit. But that basically describes how easy it is to move through anywhere. And then you see a bunch of green dots. We call that the source population. So imagine you had a bunch of animals that lived in all those green dots, and they were going to move across this resistant surface. And they prefer to move to things that are dark and avoid things that are light. The dark things are low resistance, and light things are high resistance. So then if you connected all those green dots, in the best way to connect them all, you get a network of corridors or pathways. And that's what you see in the color overlay. Red areas are the corridors that link all the green dots that are very, very frequently used. The lighter green, lighter blue are infrequently used. So this is what we talk about when we're doing connectivity. Connectivity is the functional linkage across the landscape. And that landscape is spatially heterogeneous, like you see in this resistant surface, and the distribution of the thing being connected, in this case, these green dots, will influence the connectivity network as well. So to take this to where we want to go, we have to say why connectivity is important. Connectivity is important for populations because populations change for only a few reasons. They change because organisms are born. They change because organisms die. They change because organisms move in, and they change because organisms move out. And connectivity can affect all of these things. You're going to die more if you can't get critical resources, or you're going to die more if fragmentation exposes you to more risks. There might be more birth in habitat that is more productive, and it can be more productive because it's more connected, allowing resources to be used across the life cycle of the species. Connectivity will influence whether animals move and interact too. So connectivity affects all the parameters of population dynamics in pretty fundamental ways. So we're going to get now to the first concrete example of some work on the topic. This is a landscape genetics study. The first things I did in my career after graduate school, long time ago, in northern Idaho. And what you see is a picture of the study animal, black bear, and on the left, a map showing triangles where we put up a bunch of hairs getting DNA from these bears, and we want to understand what drives genetic differentiation of bears in this landscape. So we sampled 200 or so, 250 or so locations. You have about 150 individual genotypes, which are located in these purple dots in the landscape. And we wanted to determine what connectivity, what drives connectivity of black bears in these mountains in northern Idaho. So we were going to then do an inferential statistical model. So we have a bunch of information on genetics. We have the genetic distance matrix. Here's a genetic distance matrix. You got each individual bear compared to each other individual bear. In this case, it's 6 by 6, but it's actually 146 by 146 because that's the number of bears we got. And the values in the matrix tell you how different the bears are genetically. 0 in the diagonal because that's connecting one bear to itself. But then the other off-diagonal elements tell you how proportionally different they are. 0.33 in the next one. Those bears are 33% different in their allelic richness, allelic makeup for microsatellite markers that we sample that are neutral and highly variable and should reflect genetic relatedness and how landscape influences genetic distance. And then you got other values in there, some bigger, some smaller. And then we can have hypotheses about why some bears have more genetic difference than others. And the most obvious, simple one is bears that are close together will be more related than bears that are far apart. So we can make a matrix of Euclidean distance, geographical distance between the bears, be able to see if that's related to genetic structure. Another hypothesis might be that there's a barrier. In this case, in this part of Idaho, you have a mountain range to the west, you have a mountain range to the east, there's wild places, lots of bears. And in between, you have an agricultural river valley, wheat fields, hillbillies, guns, and things like this that are problems for bears. So you might make a hypothesis that bears that are on the same side, a pair of bears on the same side of this barrier, might have no distance between them functionally, where bears on opposite sides have a one, which in this case is a categorical barrier. Comparing zeros and ones, like an analysis of variance framework. So that tests whether a barrier exists between two panmictic populations on either side of this valley. So then we can test a bunch of other things. What was novel in what this study did was it tested more than just barrier as a categorical thing, and distance as a gradient thing. It tested a bunch of hypotheses about how landscape features influence resistance, elevation, slope, roads, land cover. It tested a bunch of different ways that things like elevation and forest cover and roads might influence landscape resistance to movement of bears. And that works like this. Here's a resistance surface. I showed you one of these earlier. It's an introductory idea. In this one, let's say blue is low resistance and red is high resistance. And these are the bear locations where we have genotypes. And so what we do is test the hypothesis. If this is actually driving gene flow, this resistant surface, we should be able to measure the cost of moving across that resistant surface for each pair of bears, make a matrix, just like we saw for distance or the barrier model. But in this case, it's not Euclidean distance, it's functional distance, it's cost distance on this hypothesis of resistance. And we can do that for each of these hypotheses. In fact, we tested 110 hypotheses, different combinations of roads and elevation, slope, forest cover, and then did a bunch of multivariate tests with something called partial Mantel tests, which isn't particularly important how that test works. But the idea is it allows you to compare the support for each of your hypotheses. And importantly, it allows you to compare the support for the different landscape resistance hypotheses, accounting for Euclidean distance and this barrier idea as null models. And here's the result. This is a picture that shows 3 different factors that might influence landscape resistance for barriers: elevation, forest cover, roads. And the squares, the cubes, represent each hypothesis that is supported independently of isolation by distance and isolation by barriers. The redder the cube, the stronger the support. And if you look at the reddest places, they're all moderate middle elevation and high forest cover. And so by testing 110 hypotheses with a large genetic data set, optimizing and comparing with neutral null models, like isolation by distance and isolation by barriers, we come up with this as the best resistance model for black bear resistance in northern Idaho. Blue areas are low resistance, red areas are high resistance. The blue areas are middle elevation forest. The red areas are places like a town, a river, a road. Orange areas, wheat fields and lakes and things like that. And that shows, at least based on our empirical estimation, what landscape resistance is for bears. All right, so that gets us to another question. So when I first showed this, some bear biologists who worked in a different part of Idaho said that's completely nonsense. I know in my study area, bears are not structured genetically. They're just randomly mating with a slight difference between them if they are farther apart. So your analysis is wrong. But the important point is, this is a picture of my study area, right? You can see the valley, you can see the mountains on each side, you can see variation in elevation, you can see variation in tree cover, you have this big agricultural system in the middle. That's a high contrast landscape that might be very limiting to bear movement, where this is where the other guy worked. You see the difference? This place is all forest. No hillbilly there. No roads there. It's all middle elevation. So it's all basically the same. And it's all good for bears. So bears here just happily walk along wherever they want to go. And there's nothing that limits their movement. Where in this system, they are limited because it's naturally fragmented and heterogeneous. So the answer is, bears are doing something here that is different than there, even though maybe they're the same kinds of creatures who do the same things in the same place, but in these different places, they're doing different things. So we thought that was interesting, and we wanted to repeat our study in different places. So we did that same bear study, right? Testing these 110 different hypotheses, elevation, forest cover, roads. But we said, we're not going to just do it in one place in Idaho. We're going to do it in 12 places, 12 landscapes, across Montana, across Idaho. And we had expectations that we expected elevation will be a predictor of genetic differentiation only in places where there's a lot of variation in elevation, right? There are big mountains and mountains. It's all kind of flat. Elevation won't be detectable because it doesn't have differential influence. And we expect forest cover and land use to be a good predictor of genetic structure of bears and landscapes that have a high fragmentation of forest. It's all connected forest. It can't affect movement differentially because there's not a place that's good and not a place that's bad. It's all good. So you need to have a high heterogeneity for it to be limited. And we expected roads to be predicted as a factor influence of bear genetic structure when it's highly heterogeneous road network. When there's a lot of roads that can limit their movement. In places where there's no roads, you're not going to detect a road effect. And we expect a difference in elevation relationship as you go from north to south, with bears moving with more ease at middle elevations in the north, but higher elevations in the south because tree lines in the south, just as a climate gradient. In short, we found all of these things supported. So we find that the limiting factors are different in different places because of how elevation, forest, roads interact to limit the movement of bears. So then we went back to the guys who said, "You're wrong." We said, "Well, you know, you're both right. Isn't that great? It's like here." And that's fun. But then, you know, we got a little bit nervous because, you know, that was a nice, easy answer. 12 different studies showed differences, but we can explain the differences. But, you know, we thought, geez, we still only have some empirical data. We have a few hypotheses. We have one statistical method. The data could be limited. The hypotheses could be limited. The statistical method might not work or might be biased. How can we know that we have the right answer? So we said, we want to simulate this. Simulation. Simulation is really powerful because what we usually do, like I did with this bear example, you get some data. It's pattern, right? It's genes, you got some environmental variables, you got them distributed in space. And then you try to infer the process, right? Saying, okay, I'm doing inductive inference, I'm doing inferential statistics, I'm doing regression, I'm predicting a process from a pattern. And that's great, but you don't really know what the process is. You'll never know, you can't know. All you know is what process was best supported by your data with the hypotheses and methods and data you have. So you never know the answer. And that's not very satisfying. And here's another problem, right? Imagine this. Say you're a scientist. You have a question. You answer the question. But then you say, well, how many wrong answers are there to this question? Yeah, millions, billions, infinite. How many right answers? Depending on your epistemology, probably a lot less. Maybe only one. So what's the chance you're going to get one right answer out of an infinite pool of wrong answers? Even if you have good hypotheses, good methods, and good data. Probably not very good. So we thought simulation modeling would be good because that gives us a different way to address the problem. We know what a process is. Simulation model says we know what the process is. We know how the animals behave, where they're going, what drives their movements. We can control all that. We can produce the process from that, so we can then predict genetic patterns from hypothesized dispersal processes. So we developed this model called CDPOP. Aaron Langdon, my former PhD student, now professor at the University of Montana, wrote the model. It's a spatially explicit individual-based population genetic models. Animals move around in the landscape as a function of the cost between how easy it is to get from one place to another. They mate, the babies disperse, they die. The spatially explicit context is driven by landscape variability. Simulates changes in population structure, population size, genetic variation through time as a function of movement, reproduction, mortality on cost surfaces. So this gives a way to get to that pernicious question of, could we have gotten the right answer? Right? With this data set, this method, because, as we said, we had some genetic data, we did some regression, we inferred some landscape resistance patterns, but we don't really know what the right answer is. So now we can turn it upside down. Say, okay, start with the answers that we were trying to estimate, make them all real, make them true, because the model can simulate whatever you want it to, and then see if your statistical method works. So 3 different things we tested. Remember, I said we could have isolation by distance, that's A. Isolation by barrier, that's B. Well, isolation by landscape resistance, in this case, the best to optimize empirical landscape resistance model, we'll see. And then we're going to take all these dots, which are roughly the density and distribution of black bears in this place, and we're going to simulate the genetic structure that would result over time. And so we make the landscape resistance model drive gene flow in our simulation model. And what we expect to happen is, over time, a correlation should occur between genetic distance and the landscape resistance. And you should get stronger and stabilized. Now you see an A, and that's good. But we also see that at the same time, there are spurious correlations with distance that emerge and the barrier hypothesis emerge. And if you just look at those, yeah, they look pretty similar. You know, maybe it's hard to determine just by looking at that which is true. So there's these spurious correlations. And we've always learned that a correlation that's consistent with your hypothesis doesn't prove it's true. There's lots of other ways you could have gotten that result driven by something else. And we found that here. But what we had done in our studies, we did a series of partial regression tests that allow us to control different relationships, something called causal modeling. And that has diagnostic tests. So for example, if the landscape resistance hypothesis is correct and these distance and barrier things are not true, spurious relationships, we should be able to see this. We should see a relationship between genetic structure and the landscape resistance hypothesis, independent of barrier model and distance model. Right? Should be there after you account for the influence of these spurious hypotheses. And you should not see a relationship with the barrier model and genetics, the distance model and genetics, once you remove the relationship with the landscape hypothesis, if the landscape hypothesis is true. And you have the same analogous set of hypotheses, partial regression expectations for the barrier and the distance model, if they are true, right? So what we have is these 4 tests for the landscape hypothesis, partial tests, partial regression tests. So over time, you see a correlation in A, top left, increasing between genetic distance and the landscape pattern, removing the effect of the barrier model. Okay? There's an independent relationship with the landscape, not explainable by barriers. B, independent relationship with the landscape resistance hypothesis, not explainable by distance. And in C and D, there is no relationship with the barrier model or the distance model that is not explainable by the landscape model. So this gives us strong support for the landscape model, independent of distances and barriers. And this was able to be shown through a simulation that it worked. It worked almost perfectly. When the landscape hypothesis was true, we could reject the spurious relationships and affirm the landscape hypothesis. And conversely, when the distance and the barrier models were true, we could affirm them and reject the others. And you can do it remarkably with almost perfect power within 40 generations of starting the simulations, even well before equilibration of the population. Very quickly, signals emerge, give you 96% chance of getting the right answer with this method. And that gave us a lot of confidence because, you know, we went about this now 3 ways, right? We said, what's the best hypothesis of 110 bugs theory I got? And then we said, let's repeat that 12 times and understand why there might be some differences. And then let's do the simulation to see if this method actually works theoretically. And all of those worked pretty well. But we have some other things that we want to test. So, again, infinite number of wrong answers, very few right answers. Maybe we're still missing the boat. So we want to look at the same animal in the same place with a different kind of data, movement data in this case. Instead of looking at genetic differences between bears, we're going to look at the movement patterns of actual bears. So here's a bear, was GPS collared by some collaborators of mine, moved around the landscape. We can compare the landscape the bear utilized, the path the animal took, to the paths the animal could have taken within its dispersal ability, but didn't, with the same topology, right? Same path length, turning angles, everything else, just shifting it and rotating it randomly to control for autocorrelation. Then we used conditional logistic regression to predict landscape resistance from that. And we find different relationships with canopy cover, with roads, with elevation, in relation to the linearities of the relationship. But here's what we predict to be the resistance due to the forest cover and human development in the spring season and fall season. Do bears avoid houses and farms and gas stations? And at what scale do they avoid them, and how wide that influence is, from 100 meters up to 1,000-meter radius? And the late season is a lower value. I mean, late season is a higher value, showing less avoidance of human structures in late season, which is summer and fall, much stronger avoidance in early season, which is spring, and strong avoidance up to 400 or 500 meters. And in both cases, avoiding crossing roads, and roads of different categories. Forest roads, the first number of the triad here, county roads, and then highways. And this lower the line towards zero, strong in support. So roads are strongly influencing bear movement, particularly big, busy roads. The far right two examples are showing much bigger influence of highways than forest roads. And both of them are highly supported, particularly in the late season. And the elevation model. We have low elevation as preferred elevation, middle elevation, high elevation. And in late season, summer and fall, no relationship. They're not avoiding any elevations, they're not selecting any elevations. But in the spring, in the dark black blue line, very strong selection of middle elevations for movement. And so this gives us the ability to ask the same questions that we did with genetic data, but with movement data, and compare them. And heuristically, they come up with a very similar story, right? They're moving to forest cover, they're avoiding human development, they're not crossing roads and they can avoid it, and they prefer to move through middle elevation, particularly in the spring. And there's lots of reasons why the genetic structure with bears should be related to movement in the spring, because spring is the breeding season of bears. That's when mating occurs. Also is when dispersal happens. So dispersal and mating are two things that drive genetic structure primarily, and they happen in spring. And in the spring, movement is very, very closely related to what we inferred was important for genetics. Okay. So that gives us a background on some different ways to think about what drives connectivity, how you can use analysis to get inferences about movement, dispersal, and its relationship with gene flow and connectivity, landscape genetics, movement modeling, and simulation. And none of that is really talked about connectivity per se. That was only talked about how do we understand what influences connectivity. But we didn't actually predict any connectivity from that. So connectivity is a function of several things, like I showed before. Resistance, right? The colors from blue to red in that top graph. Source points, here's a point in the top, and a point at the bottom. And then the algorithm you use to connect. In this case, a least cost path between in that A panel, a least cost path. Or maybe we're not interested in a single path, maybe we're interested in a least cost corridor. All the ways that connect those 2 points that have at least a minimum amount of cost to connect. And that's you see in the C in the bottom. Still connecting those 2 points. But in many cases, we aren't particularly interested in those 2 points. We might be interested in what we call synoptic connectivity, which is the whole population's connectivity, all the bears in that population, how well connected they all are to each other. And as you see in B and D, analogous to least cost paths in B, analogous to least cost corridors in D. Instead of having 2 points connected, in this B, we have 1,000 points connected, about 500,000 least cost paths, connecting them all up, and red ones being more connected. And in D, we have the resistant kernel prediction, which looks at the amount of movement we expect through any single pixel given the dispersal of organisms from all those source points across this resistance surface. Red areas have high frequency of movement through them predicted. Yellow areas, intermediate, blue low, and this dark blue, you don't expect bears ever to go there. So those are several ways you can predict connectivity as a function of the resistance surface and the source points. So then, what do we use this for? Okay, you might recall that we did this bear study, right? We did it in Idaho, we repeated it 12 times, we did a big simulation study, confirmed the method works. So now we have some confidence in the resistance surface. That's what you see in this grayscale on the left. So we said, okay, what can you do with that? Well, we want to do some connectivity predictions. For example, bears from Yellowstone to Canada through the northern Rocky Mountains. It's a pretty big conservation area of interest, right? Yellowstone, Yukon, Greater Yellowstone Initiative, all these things. And so we have this empirically optimized, validated connectivity resistance surface. And then let's say, let's connect a bunch of locations along the Yellowstone northern border to the Canadian border. Let's say 200 in the south, 200 in the north, they all go and visit each other in the least cost path route. 40,000 or so least cost paths, but they all go through 3 different ways. So that tells us something important. It tells us that there are very strong limitations to the different ways you can move to the optimal routes from Yellowstone to Canada if you're a bear, and that's the resistance surface you follow. And in the right, you can lay those over a road network, a highway network, private lands, and identify all the places that there might be a problem for bears moving. And then mitigate those with overpasses or conservation mitigation or something else. And then we thought we should take this a little further. Because again, we aren't necessarily just interested in Yellowstone or Canada. We might be interested in the synoptic idea of connecting everything to everything else. So we take the same area, the same resistance surface, but now instead of taking 200 bears along the Canadian border, 200 bears along the Yellowstone border, we take about 5,000 bears across the whole place, put them in forest cover where they exist, and connect them all through least cost path analysis, about 10 million least cost paths. At the time, this took about 9 months on a fast computer to run, and you get this. Red areas are predicted to be highly important connectivity interaction routes, blue areas low, these gray areas in between, you don't predict bears ever move there. And then you can use that to prioritize conservation actions. You overlay that on public land, greens, light pale green overlay, and then you can overlay it on the highway network, like I did in the other example, but in this case, across the whole region, and rank them based on how strong the corridor is at every highway crossing. And that gives us spatial prioritization. And then we can look at a particular area where there's an important project. So this is Everett Hill, north of Missoula, from there, 93. At the time, this was probably the biggest highway crossing wildlife movement mitigation project in the country. These sort of blue tealy dots are locations where there's highway crossings built. And then you see this predicted corridor network for bears, and you see the numbers on these yellow dots, which are the relative rankings of how important that corridor is based on the strength of that corridor when it hits the highway. 2 is the second most important corridor in the whole of the Rocky Mountains, where it hits a highway. 3 and 4 down here, 3 and 4. So this is a pretty important place. And you see there's 2 teal dots right at that number 2, including one of them is a big animal bridge, a huge overpass, where 99% of the cost of this entire project was spent. So this analysis validated, or at least supported, where they placed that bridge. But it also showed that, because of jurisdictions, land use, land ownership, that project stopped there, that last teal dot, and did not mitigate these locations 3 and 4, which are the 3rd and 4th most important corridors in all the Northern Rockies. So they nailed one, but because of the limitation of the project, they missed 2 of the 2 most important other ones. This kind of analysis allows you to get spatial prioritization and evaluation. All right. So now we're going to talk a little bit about another animal. And there was interest in climate change effects on wildlife, the topic for this lecture. I'm going to spin a little bit of it here. But first I want to talk about limiting factors. Remember the bear story, right? Where I studied these bears in this place with lots of roads, lots of agriculture, lots of steep mountains. and found that those things limited bears, and somebody else studied them in a place where it was flat and continuous forest, and they found nothing. Well, we thought that was interesting, so we looked for other data sets about limiting factors and movement, and we found this one. Here, in this place in Wyoming, in the Medicine Bow Mountains, they had been monitoring American martins, tracking them in the snow, recording details of their movement paths, and for like 8 years in that place on the left before they harvested it. And then they repeated the study for like 5 years on the right. And we wanted to see, do martins do the same thing in this landscape that they do in that? And what's the difference and why? So we did the same kind of thing I showed you the bears. Right? It's path randomization. We call path selection function using conditional logistic regression. And we predicted landscape resistance from that. And here are the two results. Before they logged it on the left, after they logged it on the right. The predictions based on the actual movement of the martins before they logged it and after they logged it. So here on the left, you see this patchiness. Some places are dark, some places are a little lighter, and those reflect different forest cover types. So they're selecting this black stuff, which is low resistance, highly selective, and that's mixed conifer mesic forest. They're kind of avoiding this gray browny stuff, and that's lodgepole pine. And they're avoiding this white stuff, which is meadow. But okay, that's one thing. But then you can see how much they're avoiding it or selecting it. That avoidance on this scale goes about 0.47. Those are the magnitude of their avoidance, relatively speaking. Let's go over here. After the logging, all forest is black and dark. They're not selecting mixed conifer forest. They're not really avoiding lodgepole pine forest. But these clear cuts and these roads, which were put in, now are resistant. In fact, they're 20 times more resistant than the meadow division you want. 20 times. If you apply that model to this, you'd say, okay, those things are going to have a little bit of effect, but it's going to be like 0.47. It's not a big deal. 20 times more effect. Why? Because now there's roads and clear cuts everywhere. In any path a marten takes, you're going to hit one. And if you want to avoid it, you got to spend an entire day trying to find a way between and around these things. So, limiting factors. The few meadows in this system didn't limit marten movement, so they weren't strongly avoided. After you perforate it like mad, logging roads, it becomes much, much more limited. Just like the bear example. Limiting factors are very, very important. All right, so now we'll talk about landscape genetics, markets, climate change. The last few moments of our time. So, same place, northern Idaho, Selkirk, Purcell, Cabinet Mountains, same method, put out a bunch of hair snares for martens, about 700 of them put out over 6 or 7 years. There's the circles out here. And the ones that are solid are places we got a genotype of the marten. The ones that are empty, we didn't detect the marten. It's just like the bears, we get a genetic distance matrix, we test a bunch of hypotheses, we do it a little more sophisticated with better optimization. But we found a very simple story. Gene flow is a function of elevation. High elevations with low resistance to martens, low elevations, warm places with high resistance to martens. It's related to snowpack and vegetation. Martens forage in the subnivean stuff under the snow, and they're also very sensitive to temperature extremes. And that's then vulnerable to climate change. So we decided we would look at this more explicitly. Okay, this gives us an opportunity. If it's a simple relationship between climate and landscape resistance from marten gene flow, we can simulate that, right? We can simulate that. We can say, what's going to happen if the climate gets warmer? So we could say, okay, we can project how climate change is going to affect landscape resistance, and then we can apply connectivity models. In this case, this one down here, resistant kernels in the bottom corner D, and determine how landscape connectivity for martens may change over time. So this resistant kernel works like this. Since I talked about it every time, I haven't really shown it to you. So you have a resistance surface in A. In B, you see one dispersal kernel. Imagine the top of that red mountain up there. There's a mark, and it starts there, and it just walks randomly as a function of cost. It'll go farther and more frequently where your cost is lower. It'll go shorter and less frequently where the cost is higher. And so it's not a circle, right? It goes farther in some directions and it's redder and yellower in some directions. That's the probability distribution of where that martin will go as a function of cost. Red areas will go there more likely. Blue areas, less likely. The areas that are gray around it, we expect that martin will never go there. It's just too hard to get there for that martin. And then you can do that for all the martins, add them all together, and you get an incidence function. This is C. That's how frequently a martin will move through any cell in that landscape as a function of the distribution and density of martins and the resistance of the landscape. And then you can apply a threshold and make it into a patch. All this gray stuff is connected by dispersal at this level of movement. So we do that for our simulation, for our study area in Northern Idaho. In A, in the top left, that's the current situation, where martins have an optimal elevation for movement about 1,500 meters. And then we talk to our friends at the Climate Impact Center at the University of Washington. They say, well, by 2080, the amount of warming we expect is going to translate into your resistance model to be about a 2,000 meter optimal elevation for movement, based on our empirical analysis that I showed you earlier. That's F. And then roughly every decade in between, you get B, C, D, E. So then we can apply the resistant current model. The gray patch is where we expect to be connected at any time. So in the 1500 top A, you see basically three big patches, right? The whole of the cell curve to the left are connected. The whole of the cabinets in the bottom are connected. And this per cell stuff up on the corner right has some big valleys perforating it, but it's a big large patch. Now, if you go to F, let's just skip ahead all the way to the end, by 2080, projected climate change, everything's broken. Huge loss of connected habitat, much less of it, and many small isolated patches. So that allows us to apply climate projections, empirically based, empirically optimized resistance models, look at scenarios of how that affects connectivity, and map it. But what would that mean for the population? Call our model CD pop. That I described earlier. We can simulate gene flow, population dynamics in each of those scenarios. At 1,500 meter optimization, optimum elevation, up to 2,000 meter optimum elevation. And in our connectivity, we can measure the structure and how much connectivity there is in each of those. And that's what we see in this left panel. That's Frank-Stats metrics of connected habitat. The P lands, the percentage of the landscape. Initial condition, about 70% of the landscape was connected for martin dispersal. By 2080, at the 2,000 meter optimal elevation, about 35%. So like 50% reduction in connectivity overall. And then you have LPI, number of patches, and P, that's a measure of fragmentation. LPI is the percentage of the landscape covered by the single biggest patch of connected habitat. NP, how many separate individual patches there are. And you see there's not much change, so it's 1,800, and then there's a big threshold, bam, those things come apart, where LPI goes way, way down, number of patches goes way, way up, because big patches get broken up and fragmented, and then there are many, many isolated patches. So that just gives you a trajectory of landscape change in connectivity in the left, but what does it mean for the population genetically? Simulate the CD pop. You can simulate various things. We looked at heterozygosity and number of alleles in the population across that simulation time. The number of alleles declines almost exactly in parallel with the percentage of the landscape, right? About a 50% loss in allelic richness, roughly following the same curve as percentage of the landscape being lost. Heterozygosity is a bit more nonlinear, has a threshold of about 1,800 meters, just like the landscape fragmentation metrics go. So we find, based on our simulation, that as connectivity is reduced, the outcome influences genetic diversity in both allelic richness and heterozygosity in a very strong way. And then we can look at this at a bigger scale. All right, that was the local scale of our study, but then we can simulate this at a bigger scale across all the northern Rockies. Here's predicted allelic richness in the current landscape in A, and projected over the coming decades to 2080 to F. Areas that are red have predicted very high allelic richness. Areas in blue, very low. Black, no markers expected to be there. And you see a few things. In the current recent landscape, you see these big red blocks: Northern Continental Divide, Central Idaho, Southwest Montana, Greater Yellowstone. Those are the big red patches. And if you go here to F, those things are still red. Central Idaho is way impacted, much less red, much more fragmented. But Southwest Montana, Greater Yellowstone, not that much of an impact. But look at northwest corner of that map, which is Northern Idaho and Northwest Montana. All the red is gone. All the yellow and orange is gone. That place is predicted to be hugely impacted for martins by climate change because it's already on a tipping point. It's already on a threshold. It's relatively warm, west of the Continental Divide, relatively low elevation. So some climate warming pushes it quickly over the threshold of suitability for martins, according to this model. So then we can apply these kernel models before and after that connectivity, after that climate change scenarios, and look at what's changed. So if you add the light gray and the dark gray together, everything is covered by those two things. Is connectivity predicted now? Resistant kernel connectivity predicted at the initial condition. And then the light gray over the top is what remains connected at 28. And you see some squares or triangles and circles. Those are places that have either remained connected, that are important connections in circles, are things that are corridors that remain corridors. These are climate resilient linkages. And then there are places that are squares that were important linkages spatially, and now predicted to be broken because of climate change. And you can see where they're concentrated, Northwest Montana primarily, but some other places as well. This allows us to quantify spatially the impacts of climate change on connectivity and where those effects might influence the connectivity and structure of the population. All right, I have like 3 minutes. I want to give you one more example, since the bears and martins are a little old news. The new stuff we're working on mostly in Asia and Africa, lions and tigers, things like that. Tigers formerly lived across Eastern Asia from Pakistan all the way to Russia, and they're almost entirely gone. And what's orange here is where, optimistically people thought they were 10 years ago, but they're way worse than that, almost entirely gone. So we worked on both movement and genetics of tiger dispersal in India, in central India. And so we did this path randomization approach. As I showed you before, here are dispersal paths of juvenile tigers. We did this conditional logistic regression I described to you, and then we did the same thing I described with the martins and the bears with landscape genetics, and asked the same question. The same thing that influenced bear dispersal in movement, and tiger dispersal and movement influenced tiger dispersal in gene flow. And we found that they did. And here's a resistance model for movement and dispersal of tigers in central India, where the places that you'd think would be best for tigers, low elevation, riverian habitats, are really bad. And the places that you might think are not good for tigers, really steep escarpments, rough, unproductive locations, are low resistance. And that's because in this place, what dominates tiger habitat selection movement is people. There are lots of people in India, and they live in these valleys along these rivers. And the only places with remnant natural vegetation, any kind of prey-based, and any kind of place a tiger can find refuge for people, are in these really marginal, unproductive escarpments that are blue. And so then we were able to compare our population model and our connectivity model, put the two together, because for conservation, you need to protect core populations first and foremost, because more important than connectivity is core habitat and core populations. And so we had models from our genetic data that affected population size across space, and that's just on the left. In the gray background underneath them, you see tiger reserve in these gray patches. tiger populations are centered on these tiger reserves, and how red and how big those patches are is basically the size and distribution of the tiger population in central India. And then on the right, we have this incidence function from our resistant kernel modeling from those optimized resistance surfaces about where that tigers would predict to move. The two of them together gives us a complete picture for tiger conservation strategy, where we should protect core populations and what linkages between them are most important. The two together give us the full picture of both population and connectivity for tiger conservation to develop scenarios to preserve tigers in the future. Now, I believe I'm supposed to stop now, right? Or should I continue? Well, if you're patient, I got more. Because I got another animal I want to talk about. Clouded leopard in Borneo. Borneo is pretty cool, but it used to be cooler because in 1950 it was entirely primary tropical. It was awesome, but it's not as awesome anymore. More than half of it has been deforested, turning into rice paddies, but primarily palm oil plantations. The most rapid deforestation in the world. And this guy, the clouded leopard, is the apex carnivore. So remember this path movement stuff I described to you earlier for the martins, the bears, the tigers, and did this for clouded leopards. It's the first study ever to put GPS collars on clouded leopards in the world. In this little remnant bit of tropical forest in the Kintamanian watershed of Sabah. You see some squiggly lines, different colors or different lines. The green stuff is tropical forest, different types. The purpley stuff is palm oil planting. You can see the story pretty clearly. They stay in the tropical forest. And in one spot, they go into palm oil plantation. And you can explain exactly why they do it. Right there at the bottom. Because there's a big horseshoe bend in the river. They don't want to swim a quarter mile. And the palm oil plantation there goes all the way to the river. So if this guy on the left wants to go and eat the wild pig on the right in the tropical forest over there, the only way to get there is through palm oil. He's going to go straight across, bam, straight line. And when they go through there, they're running. But 99% of their paths are all inside this tropical forest patch. And we use path selection function, shifting, rotating paths during conditional logistic regression, predicting landscape resistance from clouded leopard movement. We get this. Blue stuff is low resistance, right? That's the forest. And the other stuff is different kinds of palm plantations based on how thick the canopy is. It's more resistant, some of it much, much more. And then we can look at scenarios. The current situation in A, here's a resistant kernel model. The incidence function, expected cloud leopard movement. And then we look at scenarios in B, if we restored all the unproductive patches of palm oil plantation, which are basically abandoned, if we put tropical forest back onto those, and then they became accessible to clouded leopards, what would happen? You can see there's more red and bigger patches, more extensiveness. And then see the same thing, but put a strip 100 meters wide of forest along the whole river, sort of a corridor along the river. You can't really see, but it is more connected. And then D, unfortunately, what's most likely to happen, all the stuff is not formally protected gets turned into oil palm plantations. And what happens? And you can see there's less red and patches become isolated and fragmented and broken up. And this gives us a way to quantify scenarios of landscape change, conservation actions, development actions, and compare them on a quantitative basis based on empirically developed understanding of how landscapes influence cloud leopard movement and a robust connectivity model, resistant kernels in this case. And so we can do the same thing, but across all of Borneo. So here's a map of Borneo. Black stuff is deforested, green stuff is forest, and yellow stuff has been deforested in the last 10 years. So I could use that with this really groovy machine learning approach to predict deforestation risk across all of Borneo, and then apply scenarios to that. And so here's the deforestation risk model. And it's different in different places because of geopolitical, economic factors. This stuff down here is Kalimantan in the southern part of Borneo, up to, you see the white line across the top. Above that white line is Malaysia. You see very different patterns because the economic and sociological processes that go on in Indonesia and Malaysia are different. In Indonesia, you see all the red stuff is right along the frontier of the forest. It doesn't go in very far. So deforestation in Indonesia happens along the edge of the forest. It doesn't go very deeply into the forest. But north, Sarawak and Sabah, 2 states of Malaysia, you see a diffusive pattern. Lots of yellow, lots of orange, not a lot of blue, not a lot of deep core areas of low deforestation risk. And that's because industrial logging is much more widespread in Malaysia. They build these big networks of roads, and Chinese, Japanese, and American companies go and harvest vast areas, where in Indonesia, they're just mainly eating yuccas. So you get very different patterns of deforestation risk. And then you can project into the future with these resistant kernel models for cloud leopard movement. Now we optimize with the movement data. D is 2010, D is 2000, E is 2010, F is 2020. And you see a change, habitat loss, habitat fragmentation, and accelerating effects, threshold effects in the recent years. And you do a similar thing with this network of least cost paths, and that by 2020, the network is much more constrained. Lots of its branches are cut. Much less overall connectivity predicted across Borneo. So that will be the end. And here's a picture I took on the shores of Chobe River in Chobe National Park in Botswana, of the monkey watching the sunset, which was cool. But just downstream of that place was this. And sort of it's a metaphor, thresholds, limiting factors, collapse, like waterfalls. And biodiversity extinction in the world is kind of like this, right? The whole planet is in a biodiversity crisis driven by habitat loss, influencing and exacerbated by climate change. And so some of these methods give us tools to understand and predict, optimize solutions to mitigate that. Combining empirical data analysis, genetics, and movement, simulation modeling, scenario analysis to optimize conservation plans. Showing a few examples for different animals in different places. So I'll stop. If you have questions, we can talk about it. Yes.
How did you predict the deforestation based on past 10 years deforestation?
Yeah, okay. If you assume the same relationships that were driving deforestation in the last 10 years will be driving deforestation in the coming 10 years, you can do a bunch of spatial statistics about where did deforestation happen, what was the neighborhood of influence, distance to roads, topography, socioeconomic factors, put into a big machine learning model, and it will spit out a probability. These places would be highly likely to not. It assumes that the same relationships, economically, socially, political, will continue, which may be not true. But if they do, it's a good prediction. Yes.
So what are the limitations of your models?
All models have lots of limitations. And that is in fact why in the bear example, for example, we asked the question two ways, right? So here's a rhetorical answer to your rhetorical question. So how many ways would it be wrong in science? How many ways are there to be wrong using independent data and different methods to get the same exact answer? Way fewer. I mean, like geometrically fewer. So that's why we always want to use empirical data to optimize a relationship. Expert opinion is worth about as much as an expert. Good data. Okay. Use good data analysis, test it with multiple methods, more independent data sets, the simulation. And if you are triangulating to the same place, you're probably pretty good. Science changes, science grows. The stuff I showed you bears, we don't do that anymore. We do a lot more sophisticated optimization. So methods are improved. Still good.
So the deployment change impact assessment. What impact will that create? Have you used another parameters?
So
Like rainfall or snow?
yeah, in this particular example, we used empirical data analysis that was related to elevation. The elevation in the system is a proxy for temperature. Right? And so we assume that that relationship will carry in the future. May or may not, there may be thresholds that would be a factor involved. But in that case, we do a bunch of other fun stuff with other variables besides just temperature, lapse rate. About elephants?
Yeah.
Yes. With you. In different places. We work in Botswana a lot, in South Africa, Zambia, Zimbabwe. Projects of any other questions?
I have some questions about scale.
Scale, I love it.
I feel like when you're developing most of these resistance models, the scale is relatively localized, it's small. And then you extrapolate out, and correct me if I'm wrong about this part, then you extrapolate that out to these larger things.
Yeah.
I guess I have two questions. One, is there a limitation to extrapolating out from kind of a local scale? And two, is there a reason we can't make these resistance models at a larger scale in the first place?
Yeah. Very good questions. And you might recall in our bear study, we said, well, we didn't really trust this one answer, so we repeated it 12.
Right, I do remember that. Yeah.
But that costs millions of dollars.
Okay.
Okay. You can't always do that. And you can't do that at all with tigers and cloud members. You don't have the data. So you can't always get the data you want. So sometimes you have to extrapolate. But you are absolutely right. Because of this limiting factor stuff I was talking about, I just argued the first half of this talk about how there's idiosyncratic local limiting factors, and things are different in different places. And then it would apply it across the whole area. So sometimes you have to do that, but it's not ideal. It's better to develop it globally, like the bear example, it would be better replicated study like that, but they're hard to do. They're expensive.
So question, but I want to rephrase it. If I can sample 100 individuals for the same cost at like a much smaller scale or at the scale of the species range, what's the advantage?
That's a difficult question.
It's the same question. I'm trying to get.
It's a difficult question because generally, broader the better. Right. But if you have 100 points and your range is, let's say, Eurasia,
Yes.
they're going to be so far apart that they're going to be genetically independent, probably.
Entirely.
Yeah. So there won't be any signal to understand why they're different because all the spatial dependence is at a finer scale. So broader is better, but you need to have it dense enough that you have spatial dependence at the scale of distances.
So if we were sampling at a much larger scale, we might be able to look at matters related to kind of historically species.
Bigger the scale, the deeper the time.
Got it. Cool. Okay. Cool. That's really interesting.
It is 5:05, so we're going to go ahead and transition to the discussion. I should get up here so folks on Zoom. Folks on Zoom, if you guys want to attend the discussion, I know there wasn't a ton of time for questions. Go ahead and join that Zoom link. Scott's putting it in the chat right now. And for everyone else, we'll take a 5 or 10 minute break and we'll head over to room 34, just right down the hall and around the corner. Thanks, everyone. Thanks, Sam. Let's give him a round of applause.